you a little bit about the work I'm just getting started at my postdoc with Peter Tippin at the University of Minnesota. And I'm going to be talking about gene expression plasticity in mutualism, and more specifically trying to think about how microbial mutualists might actually be influencing the gene expression of their hosts, and if there's genetic variation within these hosts for responses to these mutualists. And I'm going to be borrowing from a classic literature, so phenotypic plasticity, where statistically we can look for patterns in a phenotype or a gene expression that match the expectation of potentially having a genetic contribution or an environmental contribution or genetic variation for response to that environment. And traditionally in this literature, we think about these environments being the abiotic environment, like sunlight or temperature, but you can also think about that as being the biotic genotype being the environment that the organism is experiencing. So in my system, we have host plants, and there's genetic variation in those host plants, and then there can also be the environmental factor will vary is actually the genetic identity of the symbiont that's interacting with that host plant. And then you can imagine there being genetic variation for the host plant for response to that biotic environment. Okay. So the system that I'm going to be asking these questions in is actually a classic mutualism of Rhizobia and Medicago truncatula. This is a resource-based mutualism where the plant is providing carbon and resources for the rhizobia, and the rhizobia is converting nitrogen into a usable plant form. Now, this is a mutualism that's been extensively studied, in particular because it's a horizontally transmitted mutualism. So the relationship between these symbionts and the host plant is recapitulated every plant generation from the soil. And so there's been a lot of work actually digging into these signaling pathways that happen where the rhizobia is actually recognizing and the plant are recognizing each other. So there's been a ton of work here, but as an evolutionary biologist, I'm actually more interested in what's actually happening in these late stages of nodule development once that potential for that symbiosis has already been established, because this is the genetic variation that potentially evolution will be able to act on. If you can't create that symbiosis in the first place, it's essentially a dead end. And in traditional literature and theory for a long time, there were predictions that we wouldn't actually expect there to be much genetic variation in mutualisms because they might be highly constrained. But we know from large amounts of empirical evidence, including in my system, there's actually a lot of genetic variation for responses here and differences in fitness of these plants. So each of these lines is a different host plant. If you grow it with different types of symbionts, there's actually differences in fitness that are happening here. And so people have looked into, this is supposed to be nodules, have looked into this and tried to see if there are nodule phenotypes, morphological phenotypes, physiological phenotypes that are associated with these changes in host fitness when we combine them with different symbionts. And what we see is there are differences. But what we were interested in doing is looking into potential gene expression changes that might be underlying these differences in phenotypes, and use that as a way to potentially find natural variation and new phenotypes that might then be underlying these host fitness differences. Okay, so we did a very simple experiment to look at this. It's an RNA-seq experiment of transcriptomes where we have 4 different plant species and 2 different rhizobial species. The plant species this is the reference genotype for Medicago truncatula. We also have this outgroup, which is actually a sister taxa, which is used for transformation work in the system. And then we picked 2 other genotypes that span the phylogenetic diversity within this species. We've got these 2 different rhizobial species. And what we did is grew them in a greenhouse. We harvested nodules at 14 days. We have 3 replicates. We pulled out the poly-A RNA, so we're looking at the host gene expression patterns here. And we have a pretty standard pipeline. And if we look at all the genes that are actually expressed within these nodules in a principal component analysis of their gene expression, it falls out very much by phylogeny. So if you look at this PC1, basically these purple and orange are the ones that are phylogenetically most closely related. As you would expect, their gene expression patterns fall out most closely. Then we've got things that are more diverse, and then we have that sister taxa. What you can also see from this is that symbiont identity is also differentiating these samples, such that these triangles, which are melalotai, are always up and to the right in PC space. So what we can then do is we can actually use, look for those differential expression patterns and patterns of genetic variation and environmental variation and G by G based on this data. So what we used was DE-seq2, which is a negative binomial GLM. We only tested genes that passed a certain expression level threshold. And we basically had two criteria for putting genes into each of those different categories. The first was using a false discovery rate of 0.05 and using these likelihood ratio tests in these DE-seq models. The second thing we did was actually impose a log fold change criteria as well to basically try to filter out things that we think are having a big enough effect size to truly matter. Now, in order to do that, we actually, instead of pulling all of these genotypes into one model, we did 6 different pairwise comparisons between each of these genotypes, because that way we can actually look at log fold changes between pairs. Okay, so this is what the model looks like. These are the potential patterns that we could see. We're using likelihood ratio tests. And on average, across each of those 6 pairwise comparisons, what we see is, not surprisingly, we see a lot of genes popping out as being differently expressed in our different hosts. Okay, so symbiont's not influencing these, but they're different between the genotypes. Surprisingly to me, actually, we also see a lot of genes that, regardless of the host, are responding similarly to these different symbiont identities. And we see 1,900 of those. Even more surprisingly, given our lesser power we have to detect them, we also see 1,600 genes on average for each of these pairwise comparisons that show evidence of genetic variation for response to those symbiont identities. Now, one of the other reasons we did the pairwise comparisons is what you can also do is we can be even more conservative, and we can say, well, those genes for which we have evidence in 3 or more of those contrasts are those for which we have pretty good evidence that they're actually varying by the G by G, essentially. And so here's an example of just one of these genes that meets this criteria. We have 1,100 of them. And you can see that, so these are the different hosts, and these are the different symbionts. And you can see there are kind of 2 ways that you can get this G by G. One is the same slope, but different magnitudes, like the orange and the green. Another option is having a crossing reaction norm where the plant is actually changing its gene expression directionality completely in response to this symbiont. And surprisingly to me, when I categorize these 1,100 genes according to these relationships, what we see is about 30% of our contrasts have that same slope and are just being upregulated in response, whereas 70% of them are actually showing complete different, like, opposite patterns of regulation in these different types of nodules. Okay, so here's a really complex way of looking at this, but I want to basically show you what these clustering of the gene expression patterns of specifically these G by G genes. And so the first thing to note, okay, so each of these columns is a different sample. These are all G by G genes. I'm showing basically relativized expression in comparison to the overall mean. So the reds are higher and the blues are lower expression. And the first thing I want you to note is that the reference genotype, so this is actually clustering together and away from the phylogenetic expectation we would have of having similar expression to its most closely related relative. Okay, so HM101, the reference genotype for the species, is doing something totally different in these genes. And that's in contrast to the overall nodule genes that we saw. Another thing to note is that all the other genotypes are falling out first by their symbiont identity, And then they're falling out by the phylogenetic relationships we would expect to see. So there's a lot of stuff going on here. We're just starting to dig in to what some of these kind of patterns might be. But a really quick first pass thing that we can do is just look at Go terms for these and look if there's any enrichment in genes that are functions that we would expect to potentially have to do with that relationship between the plant providing carbon for the rhizobia and the rhizobia providing nitrogen. And we see a lot of things popping up that have to do with sugar transport. Remember, that's what the plant's providing as its benefit to the rhizobia. We see a lot of other kind of interesting things. I don't want to belabor this, but this is just a first pass. If anyone has any suggestions for where we can go from here, I'm all ears. So in summary, kind of what I've told you is surprising to me, we actually see extensive transcriptional variation in response to these symbiont identities. We see this at a very late stage of the symbiosis, 14 days after these nodules have actually been established, which I think is interesting. We see a lot of potential genetic variation for this. That reference genotype is doing something totally different from the other genotypes and even the sister species. We see that opposing reactionomes are really common, and we potentially see some hints that these might be things that actually have a functional role in that symbiosis. Lastly, I just want to say that this is very preliminary and we're just getting started. I haven't shown you anything that indicates that these are necessarily adaptive changes in gene expression. I would say that the fact that we see that they're not falling out by the phylogenetic relationships that we would expect suggests maybe something else is going on, and that HM101 has actually been domesticated, so that could potentially be driving some of those differences. We have data sets we can start to look further into this with, looking at GWAS candidates for nodule things with 240 of these different plant host genotypes, and we're starting to look using population genomics tools at these 240 different plant genotypes using these G by G genes as potential targets so we can look at patterns of selection. Albeit, we don't necessarily know if the patterns of selection we'd expect to see on these genes are easily detectable with the population genomic methods we've got. The last thing to say is just coevolution is obviously where everyone wants to go with mutualisms. I haven't shown you anything that really suggests there's coevolution here. All I've shown you is that there's genetic variation in the plant, that they could be evolving in response to the host. But stay tuned, maybe we'll get there eventually. Great. With that, I have a lot of people to thank, and